Hello everyone. I hope you guys had a nice 2 days holidays and everybody enjoyed at least Ganesh Chaturthi and did everything what they were supposed to do. And we are here again for our classes, and let's start with revision of the topic that we did in the last class. We by far have learned many things in DNA sequencing, starting with what do you mean by DNA sequencing, starting with what is the principle of DNA sequencing. Then we also learned about Sanger's first generation sequencing. And then also after that, we started with learning the basics about the basic idea behind DNA sequences. For that, you have to make sure and clear some bases in the background. We started with what do you mean by DNA probes. In that, we also learned about how the DNA probes are generated, how they are labeled. Then we started with what are restriction enzymes, and how restriction enzymes can help us to cleave a DNA. What are the types of restriction enzymes? Restriction enzymes are called molecular seizers, and how exactly we can use those restriction enzymes. And secondly, means application part is yet to be done from our side. And then secondly, we also learned about how do you do the nomenclature of this restriction enzymes? How do you name them? And then what are subunits are required? What are subclasses are available for type all three types of restriction enzymes that we have? And there is one type for restriction enzyme also, but we do not exactly know the exact site, means exact recognition site that that restriction enzyme cleaves at. Means how we have seen in type 2 restriction enzyme that it cleaves at palindromic sequences. Palindromic sequences are nothing but the mirror image sequences. So that is its recognition site, and we know very well the recognition sites that can be recognized by type 1 and type 3 as well, and where exactly it cleaves the DNA. In case of type 4, we just know that um, it cleaves the DNA which is methylated or normal DNA, and then what is the exact recognition site that it cleaves, that we do not know, that sequence of the thing. So it is still under this research or something like that, and we have few examples in that as well, but will not be covering that part in this particular lecture. Okay, so we just have to know that there is type 4 restriction enzyme which is under research, and people are looking for more restriction enzymes which can be used for all this cloning purposes as well. Okay, so there are many things people are researching, and this is the most important area the research is going on, because these restriction enzymes are of prime importance. Okay, so moving on, this is just this thing, what you say, this particular slide is just the revision of all the three enzymes that we have learned, restriction enzymes that we have learned. In the sense, what are the properties and what properties does type 1, type 2, and type 3 restriction enzymes has. So nature of the enzyme, it shows endonuclease and methylase activity. Type 2, it has a separate endonuclease and methylase activity, whereas type 3, it shows the endonuclease and methylase. So most of them are having nature of the enzyme is the same. So they'll be having one endonuclease subunit and one methylase unit. Okay, so methylase unit will be methylating the DNA, and endonuclease will be cleaving the DNA. Okay, then protein structure, three different subunits it has two identical subunits and it has two different subunits. So if you look at the structure, you will find this kind of a structure in there. Then restriction requirement, it requires ATP, magnesium ions, adenosine, methionine. This one type 2 requires only magnesium ions, and here also it requires ATP and magnesium ions. Then it cleaves the site random up to 1000 base pairs away from the restriction site, at or near restriction site type 2, and 24 to 26 base pair 3-2 restriction site. Then examples, eco B, type 2 is eco R1, and type 3 is eco P1. Can anyone tell me what is the since we have seen the nomenclature in the last lecture, can you tell me what do you mean by eco in this type 2 restriction enzyme that they are talking about? What do you mean by eco
E.
over
coli,
here?
the
Yes.
bacteria.
And what do you mean by R here?
Strain of E. coli.
Yes.
R itself.
What do you mean by one over here?
The first strain they have found.
The first one to be isolated, right? Okay, so this is how it is. So this is how the nomenclature is done. I'm sure it is pretty easy for the people who are from microbiology and biotechnology because this is basic of this thing. But for chemistry people, yes, you do have to learn this and you have to know what do you mean by restriction enzyme, how what do they stand for, what do they do exactly, etc. You have to know. So next is applications of restriction enzymes. So they are used in gene cloning and protein expression experiments. Restriction enzymes are used in biotechnology to cut into smaller strands and in order to study the fragment length differences among the individuals. And that experiment is known as restriction fragment length polymorphism. And each of these methods depends on use of agarose gel electrophoresis for separation of the DNA fragments, right? So once you have the DNA fragments, you will be running those DNA fragments on the agarose gel in order to find out the length of the DNA, in order to find out what exact DNA fragment you have got, or if you want to know the base pair length of the DNA, that is all you will be doing with the help of gel electrophoresis. So, so restriction enzyme has a very widespread use nowadays, and then people are still exploring that particular part of the molecular biology where they can use restriction enzyme for their own purposes of cloning and making some different protocols or different procedures which they can use. Okay. Then further in application part of the restriction enzyme, it provides the different ways of manipulating DNA, such as the creation of recombinant DNA, most important use of restriction enzyme to make a recombinant DNA, which has endless application, which will have endless application. So as I told you in the last lecture also, if you are able to cut down the DNA which is mutated and replace it with the normal DNA, normal nucleotide, then it will be a breakthrough, it will be an edge in the molecular biology field, because if you are able to do that, then you can prevent any kind of a cancer that can actually cause harm to human body, or like it will be a really good best innovation ever that can cure cancer in some or other way. So hence we many a time for cancer treatments we follow on the genetic profiles. We very much rely on the genomics, and we very much rely on the mutation part of the DNA or RNA. Okay, so restriction of enzymes also allows for the large scale production human insulin for diabetes using E. coli as well as the hepatitis B and HPV human papilloma virus vaccines. Cloning of DNA molecule and studying the nucleotide sequence. So these are the main applications of restriction enzymes that we have to we know by far, and people are still exploring. Okay. About the insulin example, I have already told you in the last lecture, and I'm sure that one example is also mentioned in your 12th standard science book. So if you guys have not forgotten it by far. Okay, so this was it about the restriction enzyme. Very simple chapter. There's nothing much to like understand in that. If you read it thoroughly and if you have listened to the lecture nicely, then you will be able to understand what do you mean by restriction enzyme and what is their application. So from here, what I want to do with you guys is, since we have learned about what is DNA and what do you mean by DNA sequencing, and we have also learned about what are restriction enzymes. So there are two experiments in your syllabus which are related to DNA. So one of the experiments that I thought I can discuss with you guys, because we are we are here, and if we are thinking that we will start the labs or something or college by November. So once we go back, we can do those experiments. So I'm sure microbiology और biotechnology वाले लोगों ने वो experiment शायद सुने रहेंगे या फिर उन्होंने at least demonstration तो देखा रहेगा. But for chemistry people, I would be explaining what exactly is this thing. So first experiment that we have in your syllabus pertaining to genomics is plant DNA extraction. Okay, so I have this particular document with me, which I'll be showing up here. Yes, which talks about the principle and the function of each ingredient of the DNA extraction. Okay. So we have genomic DNA extraction, principles, steps, and functions of the reagents. Okay. So first of all, if you want to perform all this experiment, like you want to make the fragments of the DNA, you want to check the sequence of the DNA. The first and foremost thing that you should be doing with your sample is extract the DNA out of it. And when I'm saying sample, the sample could be anything. It could be your plant tissue, it could be animal tissue, it could be human tissue. So your goal is to extract the DNA from that so that you can take that DNA and study further, right? So DNA extraction is one of the basic protocol a molecular biologist should know. Okay, and because it's a simplest procedure, and now there are so many kits available to do the DNA extraction that it has become a very simple and easy to do procedure. Okay, just that you have to get a hands on it twice or thrice, and you should be very much thorough with that particular experiment. Okay, so I think till TY, SY, or in I think mostly in your SY or TY, you must have heard about this procedure, but you must have not done the experiment hands-on. So in our lab, we do the hands-on of DNA extraction. Each one of you will be performing this, and we generally use mint leaves to extract the DNA from mint leaves that we get from the market. And generally, see, it could be any tissue, any plant material, whatever. The procedure is going to be the same. Okay, only little bit of a difference of ingredients might be there, but the principle and the procedure and the functions of each ingredient is going to be the same, right? So I have this particular document with me which talks about the genomic DNA extraction principles, steps, and functions of the reagent. So DNA extraction from a sample is a process of purifying the DNA. Okay, so most important part that you should be getting a purified DNA, which is not having any RNA entity also in there. So when we are talking about the nucleic acid contains both. It contains DNA also, it contains RNA also. So when I'm purely extracting DNA, my goal is to not to have any RNA also in there, because see, when I'm reaching for the content which is inside the nucleus, I have to make sure that I'm reaching till that part for the extraction of, see, if I'm doing the protein extraction, maybe I don't have to go till the nucleus, because we all know that translation process happens in the cytoplasm. It doesn't happen inside the nucleus. Okay, but currently what I want to do is I want to extract the DNA and RNA. So my goal is to reach till the nucleus in order to extract the DNA or RNA. We do the extraction of RNA also. Means in labs, people do the extraction of RNA also. People do the extraction of DNA also, right? So depending on what exactly you want, whether you want a DNA or whether you want the RNA, according to that you will be changing your procedure. And how you will be changing your procedure, I will explain you as we move on with this particular set of document. The sample can be tissue, plant or animal cell, blood, viral DNA, or any other DNA containing sample. The idea of extracting the DNA is quite basic, right? Destruction of the cell membrane and cell wall in case of plant cells to make the DNA exposed and then separate it from the rest of the cell debris. So very simple, you have to disrupt the cell wall, nucleic wall, and then you have to remove the cell debris and just remain with the nucleic acid content, okay, nucleus content basically. Then in this article, we will mainly discuss the extraction of the genomic or chromosomal DNA found inside the nucleus of eukaryotic cells. Okay, we are talking about eukaryotic cells. So prokaryotic क्या रहेगा? Bacteria, etc. You can extract the DNA from bacteria as well. Only little bit of a difference will be there because they don't have a nucleus wall or something like that. So you'll just have to follow a procedure which will break their cell wall, bacterial wall. Okay. Then principle of DNA extraction, very important. So firstly, cell membrane is disrupted physically or chemically to get a fluid containing all the cell component including the DNA. So पहले तो तुम break करोगे cell wall, which will give you a mixture of everything: cell debris, DNA, RNA, proteins, etc. So the process is called cell lysis, and the resulted fluid is known as lysate. So once you disrupt the cell membrane and release the content of the cell inside of the cell into the solution, that thing will be known as, the process will be called as cell lysis, and the solution that we'll be getting will be called as lysate. During cell lysis, different chemicals and reagents are used to break down the different cell components. So what are these reagents? What are these chemicals that are used? That we'll see as we proceed further. For example, lipids are broken down by detergents and surfactants. Proteins are broken down by proteases, and RNA is broken down by RNAs. Okay? So when you are extracting purely DNA, that time you have to make sure that the solution or the chemical that you are going to use should contain a lipid breaker, it should contain the protein breaker, it should also contain the RNA breaker. Okay? See, I would like to highlight one point over here is RNA is broken down using RNAs. Okay? So once you put the RNA solution, it's a very clear solution that you have, and you literally have to put only 25 microliter or lesser than that in order to make sure that your RNA is broken down and it will not be interfering with the DNA extraction procedure. In the sense, you will be, end of the day, you will be extracting only DNA, pure DNA. Okay? Because we might be able to take care of the lipid and the protein, but we might not be able to take care of the RNA when you are trying to extract all the nucleus contents. So there are high possibilities that when you extract the DNA or DNA, there are high possibilities that your DNA will be contaminated with the RNA. So that to avoid that contamination, we always use RNAs. And vice versa is applicable for other things, in the sense, when you are trying to extract the RNA, you will be making sure that you are putting DNAs. Okay? So DNAs will cleave the DNA, and what you will get in the end of the day, end of the procedure will be pure RNA. Okay? So it's vice versa. So if you are doing the DNA extraction, you will be putting RNAs, and if you are doing RNA extraction, you will be putting DNAs. Okay? So so first thing that you have to make sure that you are breaking down all the lipids, proteins, and RNAs. Then secondly, the lysate is treated with concentrated salt solution to make the broken compounds clump together and lead to DNA freely floating in the solution. Very important. Okay. Thirdly, this solution containing lysate, detergents, surfactants, broken protein, lipids, and RNA is centrifuged to separate the clump debris from the DNA. And lastly, DNA precipitation is done by adding ice cold alcohol plus a salt to increase the ionic strength, which increase the precipitation process. So very nicely stepwise they have explained. So firstly you will be putting, firstly where is the first thing, the process is called Mm, so firstly, you will be doing is you will be breaking down the lipids, proteins, and RNA. Secondly, you will be treating that lysate with the concentrated salt solution so that all the cell debris will be clumped. Third thing that you will be doing is you will be centrifuging all this clumped debris so that you will have only DNA in the clear solution. Okay, and then uh, the fourth part that you have to do is precipitate the DNA by adding alcohol and the salt solution, which will increase the ionic strength and help in precipitation of the DNA. So a pallet of DNA is obtained upon centrifugation of the solution. Okay, and supernatant is discarded except the DNA palette. So you can really see a clear white color DNA palette at the end of your append off or your centrifugation tube. Okay, and then you have to discard all the supernatant, the clear supernatant, and what you have left with the DNA palette. Okay, and then supernatant is discarded except the DNA pellet, which remains stuck to the walls of the append of. Pellet is then suspended either in slightly alkaline solution, mostly PE buffer, or ultra pure organic particles and dissolved gases removed for subsequent DNA experiment, usually PCR. So generally we can dissolve the DNA pellet in the TE buffer or ultra pure water or molecular biology grade water, which will be same as that of ultra pure water. Okay, so you have DNA solution. This is the basic principle of extraction of the DNA. So first thing that you have to take care of is that lipid, proteins, and RNA. Then secondly, you have to remove this cell debris. Then third thing is that you have to precipitate the DNA. And then fourth thing that you have to do is after the precipitation, you will have to uh, mm, separate the make the DNA pellet and resuspend it into the alkaline, little bit alkaline solution, or you have to resuspend it into the molecular biology grade water. So there are 4 to 5 steps to DNA extraction. I hope you guys have understood. Have you guys got a clear idea about it? Yes or no? Just tell me in the chat box. Have you guys understood the procedure? Yes. Okay. It takes there is a time lag of 3 to 4 seconds, I guess. I get the messages little late. So this is the diagrammatic representation, the basic steps in the preparation of the total cell DNA from a culture of bacteria. So you have so basically this is for prokaryotes that they have shown. So you have bacterial culture, then you have pellets of cell, then you take these pellets of cell and then cell extract. Okay, these cells are removed and broken given to give a cell extract. Okay, in the sense you will be adding some buffer in it which will lyse the cell wall, etc. Then you will be removing those debris and then you will be having a clear solution which will have pure DNA, and then you can precipitate the DNA through this clear solution and then get the pellet of the DNA at the end of the day. So this is a simple procedure that they have shown in a diagrammatic form. So DNA extraction steps. So steps of DNA extraction: sample collection and preparation. Okay. So DNA can be extracted from a number of sources such as human hair, urine, saliva, tissue, plant or animal cells, etc. Each kind of sample is collected according. For example, saliva is collected by using Wharton swab. That is the very well known procedure that we are doing right now uh, for COVID-19 detection also through a Wharton swab, putting in your nose. We are collecting the virus and then we are doing a polymerase chain reaction. Means in the sense if the virus RNA is there, we are converting into the DNA and making the multiple copies of it and then reading those PCR products in order to check whether that virus is there or not, and if the virus is there, then definitely it will form the double stranded DNA and will have the multiple copies of the DNA, right? So same way, means I just told you, since we are talking about collection of the DNA through saliva, uh, using cotton swabs. Urine is also collected, frozen and frozen later on. Plant leaves, human hair are easy to be collected in non-invasive way. After the successful collection of each kind of sample, it is washed with water, an organic solution, or various buffer to remove any kind of contamination from the sample. And if the sample is bacterial cell, preparation includes keeping the bacterial culture overnight. Okay. Then taking 1 ml from the overnight kept bacterial sample and centrifugation of this sample of this 1 ml bacterial culture to get the palette of the bacterial cell. I understand that we are in your third and fourth semester, we do lots and lots of your this thing, microbiology. Okay. So for chemistry people, it is little bit if you guys are having any trouble in understanding whatever I'm reading right now, because sample of bacteria is kept overnight in order to grow, because most of the bacteria take 24 hours to double. ठीक है, तो अगर तुम्हें प्लेट पे बैक्टीरिया देखना है, तुम लोगों ने थोड़ा बहुत तो माइक्रोबायोलॉजी किया है, and if you want to make extract the DNA out of those bacteria, then you'll have to have a good amount of bacteria which you can make a pellet of and then further use. Okay. So 1 ml of bacterial culture to get the pellet of the bacterial cells. Simple 1 ml solution लेना है तुम्हें फ्लास्क में से जो तुमने रात भर इंक्यूबेट किया है, and you just simply centrifuge it and you will get the pellet of bacteria. ठीक है, at the end of the centrifuge tube. Then later on, lysis of the cell pellet is done by adding lysis buffer into it. Okay, and where is it? Table 1 shows various types of sample and potential amount of DNA that can be extracted from it. So this is the table 1. Okay. So from all this sample, how much amount of the DNA you can remove, that is what's shown over here. You can see oral swabs can give you around 100 nanogram per swab to 1500 nanogram per swab. Okay. Nanogram is a very small amount. The amount of DNA that we get in our lab is literally 0.1 gram or milligram or something like that. So nanogram is very less. So imagine that how PCR, polymerase chain reaction, is helping us to amplify such a small amount of the DNA or RNA that we'll be taking through the swabs. Okay. So the sample preparation mostly involves having a pure sample with you, pure in the sense of nicely washed, less contaminated sample with you. In case of bacteria, you have to take a little bit of a care, and same is applicable for animals or या फिर mammalian cell culture also. You'll be taking a plate of cells and you'll be centrifuging it and taking the pellet of the cells. Okay, so that is what is the sample. You'll have plant samples and another what you say, what bacterial samples are very easy to do. Animal and mammalian cell cultures would be little bit difficult, but they are, you end of the day you have to get a pellet of the cells in order to proceed further with the DNA extraction. So we have less than a minute left. If there are any doubts, we'll be continuing the same in the next lecture as well. Let me tell you one thing, that I have logged the meeting. So if you guys are not being present on time, okay, I'll be locking the meeting from today onwards, because people are logging in their at their own convenience or whatever. And even if I tell you that